Hello, and welcome to the Biotechnology and Society webinar series, co-sponsored by Harvard Medical School Center for Bioethics and the Museum of Science Center for Life Sciences. I'm your host, In-Soo Hyun. I'm the director of the Center for Life Sciences here at the Museum of Science in Boston. Thank you for joining us. This is a webinar series that brings together you, the public, with experts in the field to discuss emerging issues at the cross-section of bioethics, society, and new emerging technologies, such as the technology we're going to be talking about here today. Now, you can participate by typing in your questions in the Q&A box at the bottom of your screen. Don't use a chat box, use the Q&A box. Submit your question to me, the speaker, along the way, and we will get to those toward the end of today's webinar. So we'll first have a chat for about 40 minutes or so, me and the speaker, and then you will have a chance for the Q&A interaction for the last 20 minutes. So today's topic is how new biopreservation technology may advance and disrupt the organ allocation system. Our guest today is Susan Wolf. She is the McKnight Presidential Professor of Law, Medicine, and Public Policy. She's the Farragut Baker Daniels Professor of Law, Professor of Medicine, and Regents Professor, all at the University of Minnesota. Susan, it's great to have you here with us today. I'm really curious to get your thoughts as a bioethicist, a leading bioethicist, and not just bioethics, but also in the law, about your ideas, your views of how biopreservation technology could advance and also disrupt the organ allocation system. Maybe before we jump into that specific issue, I'd like to ask you a little bit about the project in which these questions are being embedded and in which we are having this conversation today. You are the principal investigator, or one of the principal investigators of a national network project. So I want you to just tell us a little bit about that background of what is this national network? What is this project? This is something for which I've been involved with you for the last couple of years. So first of all, welcome, Susan. Welcome to the program.
Thank you, In-Soo. It's great to be here. Always great to be with you. I'm actually the lead for the ethics and public policy component of ATP Bio. And I'm going to just offer a few slides to orient people to this big, incredibly exciting project. ATP Bio stands for Advanced Technologies for the Preservation of Biological Systems. Let's see if this looks. And this is a big engineering research network funded by the National Science Foundation. So this is just a picture of the 6 institutions involved. I'm here at the University of Minnesota. Unfortunately, these look like little coronavirus particles, don't they? But that's not intentional. So what you see at Minnesota is small circles representing all the labs involved. Over at Mass General, where you are, in Boston, you see more. And then Berkeley, you see Riverside, Texas A&M, Carnegie Mellon. Lots of engineers, physicists, physicians, and then based here at Minnesota, but reaching all over the country, ethicists thinking about these new technologies. Here's just a picture to give you a sense of how incredibly exciting this is. You'll see everything here. I love here, we've got the Arctic wood frog. If you see right there in the upper left, there are remarkably a few dozen species that naturally kind of self-biopreserve. And the Arctic wood frog is one of them, where the body goes into stasis. We think of bears hibernating, but this is much more extreme in Arctic environments. And in a way, ATP Bio is an effort to harness nature, to understand that process and use it, whether it's for organ transplantation, or at the bottom you see reproductive technologies. You even see an artist's representation of the woolly mammoth, use in potentially de-extinction in the lower right, in environmental conservation, corals. We're going to focus today, of course, on organs. So I just want to, though, give you the bigger context. This is a figure we use a lot in ATP Bio just to indicate how incredibly broad this enabling platform technology for biopreserving all kinds of biological materials can be. So I've put a red circle around our focus today, transplantable tissues and organs, but look at how broad this is. Going around clockwise, to advance drug discovery, to preserve vertebrate models. In order to do research, you need to preserve, standardize your models. I talked about coral, biodiversity, food and aquaculture, to be able to biobank large supplies of tissue like skin in the case of mass disaster, even thinking about prolonged torpor or hibernation of humans for space travel. So really a wide range. This is a busy slide just to give you a sense of the very complex architecture of this engineering research center, ATP Bio. But of course, the focus today is ethics and policy, which reaches all pieces of this ERC, as we call it. And I lead the ethics and public policy component together with my colleague, Tim Pruitt, who is a transplant surgeon. He's also the past president of the Organ Procurement and Transplantation Network, OPTN, operated by the United Network for Organ Sharing, UNOS. I know we're going to talk more about both of them, and you see other colleagues crucial to our effort. And there's a very familiar picture on this screen, and Sue.
I see that.
Because you are part of this really blue ribbon group, multidisciplinary group of experts on ethics and public policy from all over the country to deal with the ethical implications of all of those applications of advanced biopreservation. Zeroing in now, this just quickly gives you a feel for how much research is going on in this really revolutionary domain of engineering, bioengineering, biomedicine. That's above the blue timeline. And below, what you can see is increasing efforts to build a community and build consensus and policy to make use of these advanced technologies for organ transplantation. This timeline just takes you up through 2019. It was the prelude to ATP Bio as an ERC when NSF first funded us. Let's zero in still further on this quick backgrounder on organ transplantation. So this slide attempts to show quickly the temperatures that our engineers are working with. So if you look at the first figure in the upper left, it's the classic, organ in a cooler being rushed by helicopter to a waiting recipient. But you see that the temperatures there usually used are above 0 centigrade. And that stresses the organ. It really confines how much time you've got, hours, to get that organ out of the body of the donor, transported and to the recipient. It's incredibly time stressed and geography constrained. But if you could suppress metabolism by going lower, lower, lower in temperatures to supercooling, to partially frozen. There's our Arctic wood frog again, and naturally who's able to attain that state. And even look at the bottom most temperatures. There's a discontinuity there because now we're talking about very, very cold temperatures to attempt vitrification, where the material becomes like glass. But throughout all of this, the enemy is ice formation, ice crystal formation. So it takes very fancy engineering to figure out how to rapidly and uniformly take something like a solid organ, which has mass. So you've got to uniformly manipulate not just the edges, but also the interior to quickly outrun ice and vitrify. That's the holy grail here, to really be able to vitrify where you could preserve indefinitely. Similarly, getting the material out of this state into a transplantable condition, again, you've got to uniformly rewarm and you've got to outrun ice crystallization. Very demanding. So here's the current standard, static cold storage. It's that organ in the box. And as I've mentioned, we've got quick disintegration, short time frame to do everything, including matching the organ to the recipient, offering the organ, getting acceptance, geographical constraints, very limited ability to test the organ, to revive or rehabilitate marginal organs. So all of this ends up with a lot of non-use of organs, and we're already way short. If you go on the UNOS website, you'll see that today, over 100,000 people in the United States, just the United States, Insu, are waiting for organs. We don't have enough organs, either in the US or globally. And as a consequence, people suffer and die. So this is another way of looking at it. I'm very grateful to my ATP Bio colleague, Dr. Insu Hyun, for a number of these slides, including this one. So right now, that's centered on Boston. He's at Mass General, so he's right near where you are, Insu. Mm-hmm. So if you talk about the gift of life and you talk about solid organs, you have a very short time frame within which to work. And that constrains you also geographically. If you could buy even 3 days, much less with vitrification, indefinite banking of organs available on demand, you could reach the globe in that amount of time, particularly if you could prevent damage to the organ in that interval. So that really is the goal. Much more than 3 days is the goal, but even 3 days would buy you a lot. So I just have 2 more slides to kind of tee up our conversation. So thinking about advances, what could ATP biotechnology get you? Particularly when you think of this biopreservation technology in the context, really, of a whole set of technologies. For example, perfusion platforms coming online that could be combined in different ways, really to revolutionize organ transplantation. So potential advances, obviously, time. Preserve organs for a longer time, maybe indefinitely, build organ banks for availability on demand, transform transplantation from middle of the night and helicopters and no time to do much to a scheduled procedure, reduce geographical constraints, deal with persisting inequities in the distribution of organs to people who live rurally, people who live distant from major organ transplant centers. There are a number of axes of inequity that are really persistent and a big problem. The National Academies has written on this. Quality, improve quality, better matching with more time, better matching of organ to recipient, facilitate testing and assessment and even rehabilitation of organs, and on the recipient side, buy time to optimize the recipient's condition. Maybe they have comorbidities, maybe there are physiological factors that need to be manipulated. Really, the ultimate goal is lives. reduce organ non-use, save lives, and along the way, improve the entire organ transplantation system. We may talk about that. What are the challenges? There are challenges. First of all, research. Research is underway. Success in research along the full translational pathway from small, first of all, bench, small animals, larger animals, preclinical, all the way out to clinical trials. Quality. We've got to assess whether organs that go through all of this engineering, they may be infused with cryoprotective agents, they may be even infused with nanoparticles, then they're subjected to the biopreservation methods. Why nanoparticles? Because that's one of the technologies being tested to pull the organs out of, if you will, the deep freeze, by using laser rewarming to activate those nanoparticles, then flush out the nanoparticles, flush out the cryoprotective agents. But if you're going to do that much to the organ, you have to have demonstrated safety, efficacy, that this is an organ that's going to last a long time for the recipient, and it is not inferior to current, more fresh means of transplantation. Something I know we're going to talk about is stakeholder input and acceptance. You want people to feel confident. You want people, whether they are transplant surgeons or patients or patient advocacy groups and the general public, to be confident and accept this technology. You need to establish appropriate oversight. I mean, this kind of very sophisticated engineering is probably beyond transplant surgeons' expertise in terms of assessing. So you need standard operating procedures, you need some oversight. We need a way of allocating. We may end up with a couple different paths for organs. Some organs will be, quickly transplanted. Some will go through a perfusion process. Some will go through a prolonged advanced biopreservation process. We need to figure out how we sort organs along those different pathways. We're going to need to build the facilities for all of this, the supply chains and the transport. And a dominant theme has to be equity. Developing approaches to this that ensure equitable access, that avoid unwanted commercialization of the transplantation system, and certainly that avoid exacerbating illegal global organ trafficking. So I just wanted to put a quick slide up acknowledging this is the tip of the iceberg, Insoo. As you know, there are lots of people who are involved in ATP Bio. And with that, I will stop sharing.
Well, thank you, Susan, for that great overview. That was very, very helpful for our audience. Let me just kind of touch base a little bit about the research that I find is just so exciting. I mean, recently, our colleagues at ATP Bio, the scientific colleagues, published a really important paper in Science where they showed that they could freeze a rat. Was it a liver? It was a rat organ, and for 100 days it was frozen. And then they did the rat.
Kidney.
Sorry?
It was kidney.
It was kidney, right. And for 100 days, and then was able to safely rewarm it without damaging it and transplanted it into other rats. And the kidneys did very well. It seemed to perform normally as if it were just kind of a fresh donation. Now, what was remarkable about this advance is that they infused the kidney organs before freezing. They infused them with these little iron nanoparticles, which then were spread throughout the organ. And then, when they were ready to rewarm it, they used an electric charge to kind of get the little nanoparticles really flipping back and forth very quickly. And it very quickly and evenly rewarmed the organ without damage, and that's kind of a major innovation. And so the idea is that technique of rewarming with the nanoparticles and flushing them out before transplantation could be used for a host of many other organ types for humans. And so, there's further testing to be done, but that was really quite a remarkable paper. In your mind, so that's a great overview. What do you think is sort of like the most urgent thing that needs to happen to prepare for this possibility that this technology could be applied to human use? What, there's so many things that need to be done between now and then. But in your mind, what's sort of top of the list? Like what really is kind of high priority?
Well, first of all, let me just say that I totally agree with you that recent article, it's by Han et al., H-A-N, with Eric Finger, John Bischoff, a number of the colleagues I thanked at the end. It's in Nature Communications, but it made the cover of Science. I mean, just as a gauge of what a big deal this is.
Yeah.
I think very high on the list is beginning to engage with the Food and Drug Administration and other regulators in this space to begin talking about how this whole innovation tranche will be overseen. Right now, Insoo, when you look at the organ transplantation system in the United States, as you know, it's kind of a crazy quilt of all sorts of authorities and regulators, even if you just look at the federal ones. It's the Food and Drug Administration looking at parts of it, like the cryoprotective agents and the machines. The CDC worries about preventing the transmission of pathogens and diseases through organs. All sorts of pieces of the Department of Health and Human Services are in on this. We've talked about UNOS, we've talked about OPTN, the Center for Medicare and Medicaid Services, CMS. I mean, I could go on. I could give you the total alphabet soup of all this. But remarkably, what is not covered there is who is assessing the quality of the organs themselves. Okay, that makes sense when the organs are essentially, these biological materials taken from one human being who generously donated and moved rapidly to another human being. But when, in fact, we're talking about very sophisticated engineering manipulation and prolonged preservation and change of state for organs, I think they start to look like a combo. They're biological, but they're kind of devices at the same time. And as I mentioned in my brief presentation, assessing the impact of all those manipulations is really typically beyond what the transplant center clinicians are trained to do. So I think what you're going to need, and our group, our ethics and public policy group, has been thinking about calling on FDA and other regulators to take really a hard look at switching gear on oversight of the organs themselves. I think that's a piece of the puzzle. The other piece that I would really nominate for central concern is the national academies have done a series of very important consensus studies on problems in the organ transplant system and worrying about inequity. Who even gets on the organ transplant waiting lists? Who gets organs? Big variation from organ transplant center to organ transplant center. One thing that we talk about in ATP Bio is, as sophisticated as this engineering is, you don't want it to become so expensive, so resource demanding, that only very few people can access it. People near the most sophisticated centers, and how will this be paid for? So I think the equity issues really have to be considered early.
That's really interesting. So do you think that this is the kind of situation where caregivers, transplant surgeons, they have to go through further education to kind of deploy these organs? I mean, is there like an additional step you think that someone would have to learn? Because that could slow down the deployment of these biopreserved organs.
Well, I think there are a lot of transplant surgeons who are also researchers already involved in this question of how are we going to make a dent in these enormous waiting lists. And, I should throw a footnote there that the waiting list understates the number of people who actually need organs, because there's some barriers to getting on the waiting list. The academies have talked about this. So advanced biopreservation is one of a suite of technologies. There are a whole set of advanced perfusion platforms. And basically what that is that can buy you some time. We may not fully know how much time, but instead of just running customary fluids through an organ as it's in the cooler, as it's being transported, you do much fancier work to perfuse oxygenated fluids through the organ to try and prevent more damage. So really, as interesting as these technologies are, they're a subset of the full range of technologies that I think organ transplant centers and surgeons are going to need to learn and wrap their heads around in order to advise patients and make judgments about which type of organs subjected to which preservation technology is most appropriate for this patient.
Right. Some may look at what we're talking about here and think there's still a huge bottleneck in just the dearth of organs that are actually donated for transplantation, right? So certainly biopreservation could help the supply issue by making organs available that otherwise wouldn't be able to, because they're not a quick match. You didn't find a quick enough match, or it can't travel that far. But still, when you look at the number of organs that are donated, the actual influx of organs compared to the demand is such a disparity there. Do you think there is an opportunity for people working in advanced biopreservation to sort of do something to encourage donation of organs? Is there any way to kind of link that up to the kind of research that we're talking about?
Wow, that is a great question. And I'm not surprised to hear such a great question, given how much thinking you do about public engagement. I do think there's an opportunity, because the more we can engage the public on first of all, the huge unmet need and inefficiencies in the system. HRSA right now is attempting to modernize the system. There really are a lot of problems. I think the more people can understand how much work is going on to make the system better, they may be inspired to participate in that system.
Yeah.
They may have more optimism that their gift is going to actually make it to a well-matched recipient and succeed in saving lives. I'd love to hear what you think about that.
Yeah, that's actually along the lines of what I was thinking. I would imagine in the best case scenario, this technology gets deployed, it kind of maxes out kind of where it could go in terms of improving people's lives, and a number of lives saved because of the limitations on actual supply of donated organs. But if there are more and more success stories about good matches, about people who otherwise would have died because they were too far away from an available organ, just story upon story of success, I think that could help turn the tide and encourage people to donate. Because as you were saying, if you feel that you are agreeing to be a donor for organ transplantation, and if you're assured that this could really go to some good use, and that there are lots of examples of how organ donation is actually making the world a better place through this technology, I think that could be very encouraging. It could be motivating for people. I think it also needs to be easier for people to opt in for the organ donation system. But maybe legislators, people who are kind of in control of those kinds of policies, would also be encouraged by the success stories. I think success is kind of what we need to first show.
I agree with you. I also think that if we can succeed with something like vitrification, if we can actually bank organs, if they're available on demand, then the potential clinical applications expand. Then somebody who's, got catastrophic heart failure, who might otherwise just not be a candidate for a heart transplant, there it is. It's available. It's banked. It routinizes organ transplantation in a way that really might also power people's commitment to donate.
So I want to remind the audience that you can submit your questions using the Q&A box at the bottom. And please help Susan and me think through some of these issues. I mean, we've been a part of this project thinking about this for a while, but we certainly welcome new perspectives and new questions that we hadn't thought about. So please submit them down below. Susan, let me just ask you, before we turn to the audience and start taking some of the questions, I mean, are you, as with any kind of technology, you've been doing bioethics for a while, and so have I. We always know that with any kind of new technology, there are always concerns about misuse. Do you see any concerns around misuse or kind of questionable use or controversial use? Because right now we've been talking about the enormous potential benefit in a medical context, to help resolve a very deep problem facing society today. But some may say, technology is always a double-edged sword, and there might be some bad uses. Do you have any kind of concern about those kinds of possibilities, and what would they be?
Well, I think in the organ transplant applications, and so the big thing that worries me is powering illegal organ trafficking. Because, if you can really bank organs, and if there's money to be made from doing that and making those organs available, I do worry about that exacerbating the problem we already have with illegal organ trafficking. I mean, there have been a lot of international efforts to combat that. But that's linked to another concern, which is, there's been decades of debate in this country about the role of money in the organ transplant system in the United States. There's a statute, a federal statute, the National Organ Transplantation Act, NOTA, N-O-T-A, which sharply curtails the role of money. It basically says people can't sell their organs, but the intermediaries processing the organs, moving them, can recover reasonable fees. But there's been persistent debate about whether some kind of exchange of value should be allowed, whether that is one way to motivate people to, donate or in other ways make available their organs. We haven't gone that direction, but these are linked questions in my mind. What in the future will be the role of money? And do we risk exacerbating organ trafficking?
As an organ donor, I have that box checked off on my license. As an organ donor, I don't know if I'm speaking for a lot of other people, but my point of view would be, personally, I would really hope that if my organs do go out into the system and there's biopreservation technology, I would really hope that it somehow, doesn't get steered into kind of a icky commercialized kind of sector, and it kind of stays in kind of the altruism track, the clinically, beneficial for those in most need. Is there anything that could be done to sort of help assure donors that those wishes will be respected? I mean, it would seem to me that those people who do the biopreservation, who kind of are the curators of these organs and help whatever this new distribution network looks like, help distribute it, that they really have to take this on as like a stewardship of valuable public resources that people have very, strong altruistic feelings around. So is there any step that could be taken to kind of assure the donor that their organs, through biopreservation, will continue to be used in kind of the most medically appropriate ways, using strict medical criteria for, the benefit of the recipients in most need, kind of that whole ethos that currently exists? wondering, is there anything we could do to sort of help preserve that ethos and assure the donor that don't worry, it's not going to get siphoned off into some kind of icky, organ trade, or you're not going to charge people too much money for access?
Well, I think we're on the verge with this technology and related technologies of a revolution that's really going to reorganize the organ transplantation system. And my guess is that the technologies are demanding enough that it invites centralization, so that it's not spread all over the place with many biobanks and facilities in competition. I think the way to keep control is to move toward more centralization than that. Selected centers, licensed centers, where there's a deliberate decision made that only those that operate probably on a nonprofit basis and that are subject to a standard operating procedures and inspection and oversight really are the banks, really are the conduits to these organs. So my guess is that that's a way to maintain quality control as well as rein in this kind of commercial impulse.
Yeah. The one graphic that you showed says that if you give us 3 days, then the organs can travel the world, right? Is there a system internationally, do you think, in place to kind of help ensure that there's proper movement of these biopreserved organs, and we don't sort of enable another kind of secondary or black market? Like, is there like an international system in place, or does there need to be one?
Well, there certainly are the beginnings of one. The World Health Organization is very involved. There are international organizations. There in Istanbul, there was an effort a number of years ago that resulted in an agreement to try to combat organ trafficking. It's really an ongoing issue. The international issues are not limited, though, I should note, to organ trafficking. There are countries where the gap between organ availability and transplant is much larger than in the United States, where organ transplantation really is barely available. And some countries, I remember there's a wonderful article that Seb Giwa and colleagues published, including some of my ATB Bio colleagues in 2017, that talked about countries at that point like Pakistan, where most of the organs retrieved were not being transplanted inside that country. So there are really big issues with fairness and equity in access and distribution.
Yeah, that's a really interesting point. I mean, some people may worry that organs that otherwise would have kind of gone locally end up going to far off places, and the feeling of, sort of we're not using our local resources to help our local patient populations. There's that kind of possible dynamic that could get introduced, right?
Yeah, I agree.
Well, at this point, I want to see if there are any questions that have come in that we could start taking on. So Sarah Winslow is here with me, and she'll read off some questions.
Yes, so this is touching on what you just talked about. So in America, we have food deserts and varied access to resources, largely dependent on area, socioeconomic status, etc. How can we make sure that these types of technologies are not worsening that wealth gap?
Well, I think that's a great question. And the most recent report from the National Academies, it was last year, 2022, on organ transplantation, focused on equity, focused on the equity concerns. So I think this is really front of mind. It's unfortunately linked to the fact that we don't have universal healthcare coverage either in this country, which is a barrier to all sorts of access to medical interventions. So I think, it's going to require real work with the payers, including CMS as a major federal payer, to make sure that these organ transplant options are available, broadly available, and it's not limited only to people who can pay cash on the barrel and afford enormous rates. It really needs to be integrated into our healthcare system.
Yes. So going back to what you were all talking about organ donation, there were some comments of people who are hesitant because they don't know where their organs are going. There's also this question about having an opt-out system for organ donation instead of an opt-in system, and how that would have ethical priorities that you've already discussed so far.
Well, there are countries that do have an opt-out system, and that's been debated in this country. I think it's a tough sell, realistically, in this country. But there have been, decades of efforts to think, how can we improve this? Required request. When a loved one is dying, should there be a requirement of request that their family members consider if they have not already made the donation decision themselves? I do think, back to the first part of the question, and Insoo, circling back to your question about what is really going to get the public fired up about organ donation, I think much more transparency about how it operates. Maybe the Museum of Science could put together a whole exhibit on what goes on with organ transplantation and what are all these new technologies coming down the pike, and how would they change it? How could they increase confidence? How can people really know where their organs go? We've, for the most part, been talking, I guess entirely been talking about deceased donor transplantation. But of course, there's also living donor transplantation where people do know where their organs are going, where they make a deliberate decision to donate, for example, a kidney while they're still alive. And sometimes, if the person they'd like to help, their friend, isn't a good match, chains of people are formed so that ultimately people are properly matched along that chain and everybody gets, a kidney along that chain.
This question is about the technology a little bit more. But it seems like this technology can work and is happening, but maybe you can also speak to the current state of the technology. But also they're asking that if this technology works and that could increase the amount of supply or the supply of organs dramatically, would that correspond to a decrease in ethical dilemmas on organ allocation?
Susan, I think that if this were to hit prime time and become a reality, I think we will get new ethical issues. I wouldn't think they would get a decrease. We might get a new set, right? So one concern I have, or one thought I have, is if biopreserved organs are slightly less good, I don't know what the research will show, but slightly less good than fresh, is there going to be kind of a tiered system of people who kind of didn't qualify for, a fresh organ, but they kind of have to get second place or second good. And that could be kind of, again, compound the access issue or kind of add a new dimension to the access issue. What are some of your thoughts about, any new ethical issues that might come up?
Yeah, I think you're right. A good analogy is reproductive technologies, where success, I think this is still true, success with transferred fresh embryos in vitro fertilization is greater than with frozen and thawed embryos. So that's an example where you do see a difference. And so the whole world of reprotech has had to think and devise, get data and devise advice on when should you go this route versus that route. I think you're absolutely right that we may find that biopreserved organs are safe enough, have adequate quality, but maybe not the same as freshly transplanted. And then there is a danger of a two-tier system. Then you've really got to come up with, well, so how are we going to allocate in a way that doesn't entrench inequities? What are the indications for going this route versus that route? For example, if you have a marginal liver that is really not a great candidate for immediate transplantation, or you can't find an appropriate match, then maybe that's an organ that should go into the biopreserved route. It possibly could be rehabilitated, further tested, deemed usable, and a better match could be found with more time. But I think you're right that you don't want it to devolve into a two-tier system, where some people get the fresh organs and some people get the organs that may have a different quality and safety profile. So I do think that's a concern.
Yeah. Susan, I kind of wish we had one of our scientific colleagues with us, because I would love to know, having a biopreserved organ on hand, would that allow for gene editing and other radical manipulations before a transplant? Because you have the time to do things. I just don't know if in that state you could actually do any, or maybe like as you're thawing it out, do genetic manipulations. Because if that's possible, then that introduces a whole other area of genetic engineering of the organs themselves.
Yeah, I'd love to have some of our scientific colleagues here to talk about that too. That's been discussed within ATP Bio. But how soon and what sort of genetic manipulations we really need them on board for. One of the pictures I showed, though, was the artist's depiction of the woolly mammoth. And it's interesting. There is an organization, for example, called Revive and Restore, that has been part of reviving an endangered species, the black-footed ferret, much smaller than woolly mammoth. And so we've been talking with them and others about what I call stacked technologies, where you might combine advanced biopreservation and cloning and de-extinction. So you and I have been focusing on organs, but there are all these other applications. One other really interesting domain that I've been working with colleagues on in ATP Bio is biopreservation, actually, of pathogens, of parasites. For example, cryptosporidium, which is a parasite that causes diarrheal illness and has been incredibly hard to work with because nobody up till now has really figured out how to biopreserve it. And so working in a lab with cryptosporidium has required passaging it through sterile pigs or other animals, really expensive, really time-consuming, hard to come up with banked, standardized strains, which is what you really need, because we don't really have effective vaccine or much treatment for cryptosporidium infection. Here's the problem. Once you figure out, as my incredibly talented colleagues at Mass General and ATP Bio have, how to biopreserve this pathogen, then you have to worry about safeguards and controls, because it's actually a pathogen that could be used by bad actors to intentionally cause diarrheal illness in a population. So it both is wonderful because it allows you to supercharge the science and hopefully come up with more treatments and vaccine, but it increases the need for safeguards.
Do you have an estimate on the number of organs that are wasted due to transportation limitations?
I don't, due to transport. But I think the best recent analysis of organ non-use is that National Academy's report from 2022. So I would look at that.
You've already touched on this a little bit, but there were a couple of questions on it to expand, I think. So do you see a role for helping to improve the quality of organs during this process? For example, a liver with mild fatty liver changes but still functioning.
Definitely. And in fact, that article by Seb Giwa et al. In 2017 in Nature Biotechnology actually talks, as I recall, about fatty livers and the ability to, defat them, if you will. So absolutely, this part of what's revolutionary about this technology. I mean, as profound as buying time is and buying space so you can move things around geographically, equally profound is the ability to intervene and rehabilitate organs so that you're not just kind of at the mercy of the insults that the organ may sustain due to just the life and diseases that the donor experienced, but you could also improve the organ potentially on its way to the recipient.
You also touched on this, but assuming that it's better to transplant organs which have never been modified, who will decide which organs will be biopreserved and which ones will not? Meaning, will there be a two-class system in the future?
Well, that's what we want to avoid. And so that's why I analogize ReproTech, where we do have, two tracks, where we do have fresh, frozen, if you will. But it's not just, oh, who can afford fresh, who can afford frozen. It's more based on research. What are the practice guidelines that govern when you go which route? Now, often it's in the same woman. Are you going to transfer fresh? Are you going to transfer frozen? But there's also donation in that sphere as well. So I think you need practice guidelines. They need to be based on research. But in the organ transplantation system, you also need system guidelines as well. As we, UNOS, OPTN, they're all involved already in formulating guidelines. We will need them to formulate guidelines for this.
Susan, I'm curious to get your thoughts on this. Do you think organs that are retrieved will be biopreserved as a default or not preserved as a default? I'm kind of curious to kind of know like that decision of biopreserve it now or not. Who makes that? And that would have to be kind of maybe planned out in advance. I'm not really sure, like that triage of whether it's going to be undergo the preservation or not. Like who would be in charge of that?
Well, it's hard to imagine that in the short term because there's such lack of organs that, really every usable organ, that's available, there's a potential recipient for that organ. So I think initially we will be talking about more marginal organs or organs where for some reason we can't identify an appropriate recipient right now through the matching process initially. I mean, if your question, Insoo, is can I envision a future where all organs go through biopreservation? I don't know. That's kind of hard to imagine. But I think what we will see is multiple tracks. We haven't talked yet about xenotransplant, involving animals. But I think there's such pressure on the organ transplantation system that there's really breakthrough work going on in the domain of xenotransplantation too. And, genetic manipulations in that sphere so that an organ that may be grown in, for example, a pig, but with the genetics manipulated so that the human recipient's body won't instantly go, pig, and reject. I think we'll see that area grow as well.
What do you think the time frame looks like for translating the success that has had in rats into human trials? When do you predict the technology would be approved to use in patients? And then another question kind of on top of that is, what do you think the consent process would look like for research and technology on human subjects?
Wow, great big questions. Well, I'll say that the perfusion technologies, which are very important and may ultimately be used kind of in tandem with these biopreservation technologies. Some of them are already approved and being used in human beings. With the biopreservation technologies, Insoo and I were talking about, this really exciting rat kidney study just published. I don't know. It's hard to, claim that I have a crystal ball here. And it's something that I periodically ask my engineering and research colleagues in ATP Bio. But, our engineering research center, we're in our first 5 years, and the breakthroughs have been incredible. I mean, to be able to vitrify rat kidneys for 100 days, that's what the Hanadal study reported, and then have them return, being transplanted, and return to function and making urine is incredible. That's why it was on the cover of Science. So hopefully we will be renewed for another 5 years. That's the way engineering research centers go if they succeed at NSF. And I would hope to see major progress over that full 10-year span. I mean, the translational pathway, you start with, bench, you start with the small animals, that's the Hanadal study and other studies. You progress to larger animals. There may be a role for some research actually in human cadavers. You then would progress to first in human. You need a lot of approvals to do a first in human. And then you progress to clinical trials. That's essentially the pathway.
Susan, I was wondering, from a research ethics standpoint, do you think there's anything unusual for kind of the clinical trials phase of this kind of study? I mean, clearly this is sort of going to have to be offered for people who are really in pretty bad circumstances, right? Any concerns there about the informed consent or the ethics of doing that kind of work?
Well, as you know, Insoo, there's a long history of ethically very challenging trials with people who are facing death.
Mm-hmm.
I mean, that's the story, as you well know and will recall, about artificial heart research.
Mm-hmm.
For example, Barney Clark and other famous cases where, you take a person who's on death's door and you say, you are on death's door. We have this very experimental technology. Are you willing to accept it? And, those of us, you and I, others in the world of ethics, have worried long and hard about the quality of consent that you get in that kind of circumstance. And some of those trials also, I mean, that's a different technology. The artificial heart resulted in stroke, resulted in cognitive impairment, which then makes the ethics even more difficult. I mean, a basic tenet of research ethics is not only free and voluntary consent, but the ability to withdraw at any time. So all of that is going to be subject to real scrutiny when we talk about first in human trials with technology like this.
Well, Susan, we're just about out of time. I just really first of all, I wanted to thank you for sharing all the great work that you've been doing with ATP Bio and for giving us so many things to think about. It's an exciting future, but it's also full of challenges, as you and I know. I hope that the audience learned something new today. I wanted to thank the Harvard Center for Bioethics for co-sponsoring this event, along with the Museum of Science. I want to thank you, Susan, for joining us today, and Sarah Winslow for moderating the Q&A. And I want to thank you, our audience, for taking your time to join us in this conversation. Thank you for your questions. Those are wonderful. And we're going to close here. I hope you have a nice rest of your day, and thank you for joining us.